Welcome to the third part of the 2024 analysis enhancement videos. I'm Kate Ramery and I'll be taking you through the genomic breed reference update. The genomic breed reference update came about from the rapid uptake of genotyping across many breeds. So in the graph on the left-hand side, the green bars show the number of genotypes taken over the last 10 years. And as you can see, there's been a spike in the last 2 or 3 years. This graph in the bottom right-hand side is showing breeds that haven't previously had access to genomically enhanced breeding values, but have been genotyping all the same. So the most obvious one there is the composite shedders, and they have around 12,000 genotypes taken that haven't been included in the analysis as of yet, but will be going forward. So overall, there will be just over 29,000 new genotypes included in the terminal analysis and just under 14,000 in the maternal analysis. This is actually 18,000 new genotypes with some filtering between the two analyses to make up that leftover. So previously we had 13 breeds that had access to genomically enhanced breeding values, and they were published on our website in this table on the left-hand side. And these animals were identified via their ID. We looked at breed composition within their pedigree, essentially, and where we found an animal that had more than 50% of an acceptable breed, they were given a genomically enhanced breeding value as a result. Going forward, a breed or population will be identified from the genotype itself. So we'll be looking at the relatedness, an animal's relatedness to an identifiable population based on their genes. So the colourful graph down the bottom is showing the structure analysis used to identify breeds or populations, and each colour represents a population or a cluster of individual animals based on genome patterns. So I have a couple of examples here. The first is a CT animal that has a pole Dorset sire and an ultra-white dam. Previously, this animal would not have received genomics because ultra-whites didn't have access to genomically enhanced breeding values. Going forward, ultra-whites will have access to genomically enhanced breeding values, and therefore this CT animal will meet breed comp standards. Our second example is a CT animal that has a pole Dorset sire and extended pedigree, but an unknown dam. So previously this animal would not have received genomics because we don't know what 50% of its breed composition is. Going forward, this animal will receive genomics. Almost all animals and breeds or populations will now receive genomically enhanced breeding values. So a quick look into reference populations. So a reference population is referring to a group of animals that have been both genotyped and phenotyped, phenotyped being a trait measurement. And this group of animals is used to inform the breeding values of animals that haven't been measured for that trait, but have been genotyped, or to better inform the breeding values of those animals. So genomically enhanced ASBVs utilize genomic information to more accurately inform relationships. So the inclusion of more genotypes will result in larger reference populations and therefore more accurate ASBVs. So reference populations are breed or population specific as well as trait specific. So if we were looking to inform the IMF breeding value of a white Suffolk, we'll be looking at a group of white Suffolk animals that have been measured for IMF as the reference. So reference populations need to be maintained by continuing to phenotype and genotype a variety of animals for all traits of importance. So the changes that will be seen in the analysis, as I said, we've got over 18,000 new genotypes included across LandPlan. And the biggest impact will be seen in animals that receive a genomically enhanced breeding value for the first time. I guess the group that will be second most impacted will be animals that have a genotype already included, because that reference will be shifting a little bit. And the least impacted group of animals will be those that don't have a genotype at all. So in terms of traits, the traits will be impacted the most will be those that are hard to measure, so sex limited, measured later in life, or on carcasses. So that'll be traits like IMF, shear force, reproduction, etc. So genomics increases the accuracy of the ASBV. there is an equal chance that that ASBV will move in a favourable direction as there is for it to move in an unfavourable direction. It is the accuracy that is changing. And finally, what does this mean for you? So there'll be more genotypes in the analysis, which will be great. That's more information informing breeding values. There'll be a larger and more accurate reference population. And there will be some movement in ASBVs, especially where a new genotype has been included for the first time. Thank you.